Hello, my name is Ece Demir. I am a senior environmental engineering student at Middle East Technical University. Today, I'll present you a paper published by Tobias Fischer in 2001, titled Screening for Soil Toxicity and Mutagenicity using Luminescent Bacterium, Case Study of the Explosive TNT. You can see the outline of my presentation here. I'll start by introducing you to the topic, then I'll move on to materials and methods utilized in the study. Afterwards, I'll explain and discuss the results of the study, and I'll share a screening strategy proposed by the author. Then our presentation will be concluded. Now, let's start with the introduction. The study explores the suitability of marine luminescent bacterium Vibrio fischeri as screening indicators for soil toxicity and mutagenicity. TNT was one of the most widely produced explosives, and the contamination of soils by TNT presents a problem at many former explosive manufacturing and related military sites. The TNT molecule exhibits a high toxicological hazard, where effects were reported for humans and several aquatic and terrestrial organisms. In addition, evidence for mutagenic and cancerogenic potential of the compound were reported. Now, why was luminescent bacterium used in the study? Luminescent bacteria assays are rapid, sensitive, and cost-effective screening tools. Furthermore, there is a sensitivity of biofishery towards TNT and its derivatives. The analogous appearance of reduced metabolites during the reductive biotransformation of TNT would reflect the process of detoxification of the TNT molecule, which is detectable by luminescent bacteria. The study consists of two parts. First, the determination of the bacterial toxicity and mutagenicity of a single substance TNT and its primary reduced metabolites. The second, the determination of the water-extractable toxicity and mutagenicity of soil samples taken at a formal production plant for TNT showing complex contamination. This section describes the toxicity and mutagenicity of TNT and its primary reduced metabolites, 2-ADNT and 4-ADNT. These two metabolites were analyzed as single substances because they are known to be the first stable metabolites of the parent molecule TNT, and they have naturally occurring transformations in soils. 3 different assays were conducted, all measuring bacterial luminescence after incubation with acute samples of the test substances. Acute bacterial toxicity was assessed by a short-term assay, and a long-term version of this assay was used, which would include potential effects of the biosynthetic processes. These results from the short-term and long-term assays were compared to those generated by the Mutagenicity test, where restoration of the luminescence in a special dark strain of Vibrio fischeri after exposure to the test substances indicates bacterial mutagenicity. In the second part of the study, the short-term and the Mutagenicity assay were applied to detect the acute toxic and mutagenic potential of several soil samples taken at formal production plant for TNT. For this purpose, aquatic bioassays were performed with aqueous leachates prepared from the soil samples. Leachates were preferred for screening instead of organic soil extracts to determine the worst-case scenario for the concentration of soil pore water and the worst-case scenario of potentially mobilizable fraction of water-soluble soil contaminants into the groundwater. Now, let's move on to materials and methods utilized in the study. I'll be describing 4 methods utilized, starting by characterization of soil samples. Soil samples for this study were taken from 5 experimental plots. The site for this project is located at a formal production plant for TNT in the Harz Mountain in northern Germany. From each of the 5 plots called U, B, and P, and K, samples were prepared, physicochemical soil characterization was done, nitroaromatic compounds in soil samples were extracted and determined. Soil contents of polycyclic aromatic compounds were analyzed, and heavy metals were extracted from soil. TNT concentration of the sample from plots P and K shows heavy TNT contamination, whereas soil samples from plots B and M must be considered weakly contaminated. It should be noted that chemicals with a higher tendency for sorption to the soil matrix, which is immobilization, will not be extracted at high concentrations. Contamination with nitroaromatics is accompanied by the elevated concentration of PAHs and heavy metals. This co-contamination can be explained by the special techniques employed for TNT production 50 years ago. Nevertheless, even soil sample U, which is the reference plot showing no contamination by nitroaromatics, has elevated contents of heavy metals. Thus, a certain background level of heavy metals observed in all 5 samples can be assigned to historical mining activities in the whole area. Let's discuss the preparation of acute samples and soil leachates for bioassays. Acute solutions of TNT, 2-ADNT, and 4-ADNT were prepared in water without addition of any co-solvent. No further processing of these samples was necessary before applying to the dry growth media, which is a part of the Mutatox test kit. Let's discuss the luminescent bacteria toxicity testing. Toxicity testing with Bioassays fishery was performed using commercially available kits with liquid-dried luminescent bacteria. To determine the acute toxicity of acute samples, the short-term luminescence inhibition assay was carried out. Briefly, serial dilutions of prepared samples were incubated. At the end of the exposure time, bacterial luminescence was measured using a photomultiplier. Additionally to the established short-term assay, the toxicity of TNT and its primary reduced metabolites was determined using a long-term assay. Compared to short-term assay, only test medium is altered to allow bacterial growth and luminescence for 24 hours. Besides luminescence, the growth of inoculated bacteria can be determined with the measurements of optical densities. However, as both metabolites show colored solutions and interfering with photometric measurements was expected, these measurements were only performed with TNT. Now let's discuss Bacterial Mutagenicity Assay, which is the Mutatox test. Mutatox test is performed with a special large strain of Vibrio fishery producing very low light during its growth cycle. Light production is restored when cells are grown in the presence of subacute concentration of genotoxic agents. Consequently, the increased luminescence is measured. The test system consists of two kinds of dry growth media, with and without S9 fraction. S9 fraction contains enzymes and appropriate cofactors. The Mutatox test was performed by the standard Mutatox protocol, where media is serially diluted, incubated for 24 hours, and then mutagenic response was determined by the measurement of bacterial luminescence at 16, 20, and 24 hours using a photomultiplier. The qualitative interpretation of the experimental data was done by the modified two-fold increased rule, and the quantitative description of the mutagenic response was done by identifying the dilution step showing the highest luminescence as the peak concentration, MPC. And the MPC is calculated on the basis of the results from the chemical analysis of the samples. Our discussion regarding the materials and methods of the study are done, and now we can discuss the results. We will be discussing the results of the first and second part of the study. The results from short and long-term toxicity testing of compounds are summarized in Table 2. The effective concentration, EC, is used to evaluate toxicity. EC means concentration that produces 20 or 50% change in the response. EC20 and EC50 values for short-term assay reveal a typical sequence of decreasing toxicity. These identify TNT to be the most toxic compound, followed by 4-ADNT and 2-ADNT. EC values for TNT are in the range of literature studies, but for both metabolites, about 2 to 4-fold lower EC values were determined, indicating higher toxicity. Long-term assay reveals a clear difference in sensitivity between the two assays. EC50 values from long-term assay are about 10-fold lower for TNT and about 4-fold lower for metabolites, indicating this method to be more sensitive than the short-term assay. In addition to luminescence, the inhibition of bacterial cell growth caused by TNT was determined in the long-term assay by measurement of optical densities. The EC50 value of TNT is significantly lower than the corresponding value generated from luminescence measurements, thus indicating that cell growth is a less sensitive toxicological endpoint in the long-term assay than luminescence. Results from bacterial mutagenicity testing of the single substances are presented in Table 3. All three compounds were identified as mutagenic, with the order of decreasing mutagenicity being TNT, followed by 2-ADNT and 4-ADNT. Results from toxicity and mutagenicity testing of soil leachates are presented in Table 4. Based on EC20 values, the order of decreasing toxicity and increasing EC20 was determined to be K, P, M, B, U, K being the most toxic, U being the least toxic. EC values extracted from the leachates was compared with the EC20 value for the single substance of TNT. These values clearly shows that concentrations of TNT in the leachates explain almost all of the observed leachate toxicities. Mutatox results determined 3 of the 4 leachates as mutagenic. Maximum peak concentrations generated from leachate concentrations of TNT for these samples are in the range of the mutagenic responses observed from the single substance TNT in aqueous solutions as described, and thus suggests that TNT was the predominant mutagenic compound in these leachates. Now that we know the results of the study, let's discuss them. The discussion will be done in two parts, for single substances and then for soil leachates. The order of decreasing toxicity with the parent molecule TNT as the most toxic compound and broad primarily reduced metabolites showing lower inhibition of bacterial luminescence was confirmed. The resulting EC values of the short-term assay for TNT are in the range of literature studies, while for both primarily reduced metabolites, EC values were lower, indicating higher toxicity of these compounds in this study compared to literature. However, a comparison of the corresponding EC values of the presented study indicates long-term version to be more sensitive than the short-term assay, describing the increasing toxicity of these compounds with an increase in exposure time. From optical density measurements in the long-term assay, it can be stated that luminescence was adversely affected by a longer exposure time, while cell growth was not, compared to short-term. From the experiments conducted in this study, the following order of sensitivity of experimental endpoints towards TNT toxicologically is derived. Luminescence in the long term is the most sensitive, followed by cell growth in the long-term assay and luminescence in the short-term assay, as you can see in this graph. The order of decreasing mutagenicity has been determined to be TNT, 2A-DNT, and 4A-DNT. The author suspects that the long-term toxic effects of the nitroaromatics in terms of growth and luminescence inhibition may inhibit the elevated light output of bacteria in the Mutatox assay. Overall, one may conclude that the Mutatox assay can detect, but tends to underestimate, the inherent mutagenic potential of nitroaromatics. Now let's discuss the results of soil leachates. Experimental results indicate that almost all of the water-extractable acute bacterial toxicity of the samples can be explained by TNT alone. The several co-contaminants identified by chemical analysis of soil samples played only a minor role due to mobility. S9 metabolizing enzymes was used in some samples of Mutatox to support bacterial growth. However, a restriction remains for the application of metabolizing enzymes S9 in soil leachate testing, as there is evidence for possible false positive responses due to natural substances from soil which may be enzymically activated. Mutatox assay performed with the leachates generally seems to be a suitable way to detect water-extractable mutagenic potential in soils, and there is evidence for extracted TNT being predominantly responsible for mutagenic potential of soils. Based on the results of the study, the author proposes the following strategy for the evaluation of soil toxicity and mutagenicity based on luminescent bacteria assays with leachate on TNT-contaminated soils. Before remediation, performance of short-term assay to determine the initial water-extractable soil toxicity and identification of the major toxic compound should be done. If the results reveal TNT to be the major toxicant, then the Mutatox will not be necessary, as a positive result is expected. During remediation, the periodical performance of short-term assay to observe occurring changes in water-extractable soil toxicity should be done. And after remediation, if the acute toxic potential of leachates as determined by the short-term assay is significantly reduced, or if there is no toxicity remaining, the Mutatox assay should be performed to confirm similar reduction of the water-extractable mutagenicity of soils. To conclude, the proposed leachate-based screening strategy for soil toxicity assessment using the short-term assay in combination with chemical analysis provides a powerful tool for the identification of major toxic compounds in soil. This approach may be specifically applicable for the case of bioremediation of TNT-contaminated soils. Thank you for listening to me. If you have any questions, you can ask me. And here are my questions to you.